Right, in this lesson, we're going to take a very quick look at how DNA makes a copy of itself. We call that DNA replication. We took a look at this in detail during the cell cycle. We call this the phase synthesis. Now we're going to take a look at exactly what's required for DNA to make copies of itself. Now, we're just going to focus in on a few of the basics as we go through this. First thing to mention here is that DNA is held together by weak hydrogen bonds. We need to break those. So there's an enzyme called helicase. Note the ASE. That tells me it's an enzyme. Helicase is what unwinds and unzips the DNA molecule. When I say unwind, remember DNA is a helicase, and then unzip, very much like a zipper, it breaks those hydrogen bonds and opens up the molecule. Before you can read a book, you have to open it. Same thing applies to DNA. The second one is called DNA polymerase. DNA polymerase is also an enzyme, but in this one, it's going to add free DNA nucleotides, those individual pieces, in a complementary fashion. So if it reads a T, then it places the A opposite of it. We call the strand that's reading the template strand, and you'll see that a little bit more in a second. Now, another thing about DNA is that it is what we call anti-parallel. What that means is that the strands are read and built in opposite directions. So you don't read both strands in the same direction. And this becomes important later on in terms of how the DNA molecules can be built. So what happens here is this DNA molecule is unzipped by the helicase. So you notice that in this case, the helicase is moving up. Also notice that the ends of the DNA molecule are labeled. We call those ends 5' and 3'. Don't necessarily worry about that right now, but those are based on where the phosphate group is versus the sugar group. So the 5' end is the beginning of each of these strands. So notice that the left strand begins on the top and the right strand actually begins on the bottom. But to helicase, that doesn't matter. DNA polymerase, on the other hand, that matters. Okay? It can only build from 5' to 3'. So note the direction that this polymerase molecule has to move in to build this complementary strand. It has to begin here and build in this direction. Now that's going to be important in terms of how it deals with that replication fork later on. We call replication semi-conservative. Semi means half, and conservative means saved. When DNA is copied, you can see here that the red represents new material, while the blue represents the original DNA molecule. When you make copies of DNA, what you're actually doing is using half of the old material and adding onto that with half new material. So what this means is half of the original strand is saved when copies are made. This is where you can see how that anti-parallel becomes problematic. If you note, new strand on this molecule can be built up. And remember, the helicase is moving up as well. The problem with that is the other strand has to be built from as far as the replication fork has gotten down. Then what have to do is when the helicase unzips a little bit more, it'll have to pick up and build to where it left off. And so what you're going to see here is that you're going to get these gaps. Okay. This is called the lagging or discontinuous strand, while this one can be built without interruption. That's called the continuous strand. And what it means is we're going to have gaps on the side of this DNA strand when it's built. Those are called Okazaki fragments from the person who discovered those. And those are going to be filled in by another enzyme a little bit later called ligase. Now, normally, if we were meeting together, we would have a modeling activity. I've got foam models that allow us to build and replicate these DNA molecules. Obviously, that's not going to happen here. We're going to try and do as much of this virtually as we can. And so I've got a couple activities that are related to this exercise that I'd like for you guys to try out. And you'll find those on the sheet in Google Classroom. When you're done with that, you're going to find an assessment in Skyward. Make sure that you've completed both activities, that you understand both activities, and if you've got questions, come to my office hours on Wednesday before you take the assessment, okay? Make sure that you're taking good notes so that you have those as a reference tool as you're going through the assessment. This will be useful for you when you take the assessment, okay? So if you've got questions on that, we'll take a look at those more specifically later. In terms of speed of replication, I'm going to skip over this. I just want you to know that as we're portraying this, it looks as though DNA is just replicated in one fell swoop. That's not necessarily the case. We can replicate DNA in multiple locations at the same time. Now, in bacteria, we can do that with 2 replication forks. And notice that one strand will be built in one direction, one strand will be built in the opposite direction, and eventually they'll meet in the middle. So they'll begin on one end and finish on the other. Now, this is bacteria, a circular piece of DNA. You have 46 chromosomes, and you're going to have to replicate those in a very short time span. So in eukaryotes, we speed this process up by replicating DNA in multiple locations. The more replication forks you have, the faster the process goes. I don't want to get into too much detail about that. I just wanted you to be aware of that. Okay? So from this point, let's move on to the interactivity and science skills activity in Pearson. And remember, if you're having difficulty with those, meet me in the off hours and we'll take a closer look at that. So good luck, and we'll talk to you